Thank you. It's a pleasure to be here, pleasure to get an opportunity to share with you some of the work we're doing at Cornell in the area of big data and how we use this as a way to personalize cancer medicine. So I'm going to sort of talk about cancer throughout this talk. I just wanted to give you a little bit of background about cancer. Cancer is a collection of disease, typically sort of found in different locations in the body. You've heard about lung cancer, colon cancer, prostate cancer, and so on. So that's how in the past we've been thinking about cancer, about really the location of the cancer within the body. But it's something in common about all the cancers. They're characterized by abnormal proliferation of cells that divide and don't stop dividing and create a tumor and go to different organs and create a lot of damage. So that's the disease that we study at Cornell. Cancer is caused by mutations. Cancer is coming from one cell that acquires a mutation that makes the cells divide and not stop dividing until it creates a tumor with billions and billions of cells and with more additional mutations. The mutations that you see in cancer are in the DNA. Then the DNA of those cancer cells, and they make the cancer cells different from all of the cells that we have in our body. The mutations are coming from multiple sources. UV light is a source of mutations. Smoke also creates mutations. Lots of different ways by which you get mutations. Even randomly, cells, when they divide, they also create mutations, and some of them are not repaired, and that can give rise to cancer. So cancer is an accumulation of mutations in the DNA. All kinds of different mutations from single nucleotides. You see this letter ACTG. The genome can acquire mutations that change one nucleotide into a different nucleotide. And that, if it's in a bad location, a location of a gene that is important for proliferation of cells, can start the process of cancer. So what we're trying to do at our institute at Cornell is to understand where these mutations are located in the DNA. We take biopsies from cancer patients and we sequence the DNA of these biopsies. The mutations in the DNA don't really do anything. It's really when they're located in genes. The genes are coding for the machines of the cells, what we call proteins. And the mutations are actually causing proteins with a different abnormal function. That's really the reason why mutations sort of are bad, because they're causing sort of changes in the function of proteins that come out of the DNA. And so how do mutations cause cancers? These proteins that have abnormal function are basically sending signals to the cells to divide and not stop dividing. And that's really the process by which sort of cancer works. So thankfully in the past sort of few decades now, especially in the past few years, things have really accelerated quite a bit in the past few years. The field has come up with ways to block the function of these abnormal proteins. So proteins that have mutations have a slightly different shape. And people have come up with ways to block the function of these proteins by essentially inserting small molecules or using antibodies to block the function of these proteins with abnormal function. Think of it as essentially a key that goes in the lock, and then you break the key so that the key is completely engaged and is blocking essentially the lock. So that's really how targeted therapies work. They're blocking the function of abnormal proteins caused by mutations. So what's been happening is, as a result of really now understanding cancer better, we know that there are lots and lots of mutations on which you have a potential key. So what is really become important in the field of cancer is to sequence the entire DNA of cancer cells and to find all of the mutations that the cancer has, so that you can essentially combine multiple keys to block all the different locks in cancer cells. Thankfully, this is something that we can do thanks to technologies. Now we can sequence somebody's cancer genome in just a couple of days. It's a revolution in cancer treatment to be able to understand really what's causing the cancer in each individual. And what we see as a result of sequencing a lot of cancer genomes, a lot of cancer DNA, is that everybody gets a different cancer. When you hear that two people get breast cancer, if you look at the mutations that drive the cancer in those two people, they're actually very different mutations. And so what that means is that you need to treat those patients with two different ways, in two different sort of distinct ways, with two different therapies or combinations. And so that's what we've been doing in our institute at Cornell. We've pioneered new ways to sequence DNA of cancer patients by sequencing the entire DNA, all of the genes. We want to make sure that we find the entire set of genes within the cancers and really sort of understand what's causing the disease in a lot of detail. And we've been doing this now for several years. We sequence thousands of patients, and we're really sort of able to use this information as a way to treat patients in a very novel way, in a very effective way. This is an example of a bladder cancer patient, a patient that had a lot of multiple metastases. So the cancer had migrated to different locations in the body, very aggressive cancer, very difficult to treat patients with multiple tumors in this way. But by taking a biopsy from some of the tumor cells, extracting the DNA, sequencing the DNA, we were able to find a mutation that was driving the cancer in all of the tumors of that patient. And thankfully, there was a drug that would target the product of that mutation. It's what you see on the left side. What's really interesting about the mutation that's driving the cancer in this patient is that this mutation is not often found in bladder cancer. It's actually commonly found in breast cancer. In breast cancer, the drug on the left side is really what's used. So what we did in this patient, because we were running out of options, if nothing was working, chemotherapy wasn't working anymore, we treated the bladder cancer patient with a breast cancer drug. And because the breast cancer drug, would essentially target the mechanisms that are causing the cancer in this patient, this drug worked really nicely. And as you can see on the left side, the patient had multiple tumors. After treatment, all the patient's tumors were gone as a result of really understanding the drivers of the cancer and targeting them in a very sort of individualized way. And what's really interesting, and going back to sort of a redefinition of cancer, we see the same mutations in multiple tumors, and irrespective of where the tumor is located in the body. This is a tumor here from a uterine cancer, where we found the same tumor that we would see in bladder cancer and also in breast cancer, but at a different location. And also, we treated the patients with the same drug, and the patient responded really well to this drug. As you can see on the right side, the tumor is gone. So really, it's a redefinition of cancer away from where the cancer is located in the body to what kind of mutations are actually driving the cancer in the first place and how we can target those mutations to treat cancer. It's a novel way to think about cancer that's very effective. So this is what we've been doing in the past few years. I just wanted to give you a little bit of insight about how we think about cancer and big data. Cancer has become a big data problem because when you sequence somebody's genome, you generate about 1 terabyte of information. That's how much DNA we have to sequence to be able to find mutations. So it's really becoming a big data problem that we have to address in multiple ways. But at the institute, we not only sequence the DNA, but we do additional things. So for example, we take cells from the patient, we grow the tumor cells outside of the patients in dishes, and we make mini tumors, essentially what we call avatars. The idea is to try to make a lot of mini tumors that are copies of a patient's tumor, and we can try different drugs in these tumors and different combinations and see what works and what doesn't work. It's basically trying to get a functional readout of a genome. These mini tumors that I was just telling you about is what you see on the right side. These are all of these different dots. This is a mini tumor. They're all coming from one patient, and you can try different drugs on each of the mini tumors and see if they work, if they can kill cancer cells effectively. And so we're using robotics as a way to test multiple drugs. We can test up to 1,000 drugs on one patient. Basically, it's like conducting a clinical trial for each individual patient. We basically know what works and what doesn't, and then we can treat the patients based on that information. So we're really now excited to expand this concept of precision medicine and genomic, sort of sequencing outside of cancer. We're thinking broadly that, in 10 years, maybe 20 years from now, everybody will get their genome sequenced. When you enter medical systems like ours, you get a genome sequenced. The genome sequence goes into the medical record, and it's used throughout the entire life of a patient from beginning to end. Genomes can be used in just so many ways, even beyond cancer. They can be used for diagnosis of conditions. I'll show you an example in the next slide. They can be used for pharmacogenomics, which is to give a right medication dosage to individual patients. Different patients should get different doses of medication based on the genome. We know that for sure. Now we have the opportunity to do this with our patients. but also really think about prevention, think about detecting patients that have a high risk of developing breast cancer, for example, potentially 10 years, 20 years before they actually develop the disease, and sort of putting them on a path towards decreasing the risk of developing these conditions. We can do this using precision medicine, using genomics, and these are things that we are looking to do at Cornell. This is an example of utility of genomic sequencing outside of cancer. This is a patient that we sort of cared for at Cornell that had lifelong neutropenia, basically a lot of immune sort of cell activation that gave rise to essentially a lot of ulcers, a lot of blisters, in the mouth of his patients as well in other places. And the patient had had this condition for his entire life and no way to resolve this condition. We sequenced the genome, the DNA of that patient. We found a mutation in the gene called MEV, which is really interesting for us because finding that mutation enables us to actually get a diagnosis for these patients. When you see this kind of mutation, you know that the patient had a Mediterranean fever diagnosis. And that allows us to, once you have a diagnosis, to find the right drug to treat the patients. Without a diagnosis, you can't treat the patient. And that led us to treat these patients with colchicine, and that led to resolution of the disease. The patient that had a condition for 47 years, undiagnosed, thanks to DNA sequencing, we were able to help this patient. So we're sequencing the genome of other types of patients, for example, Alzheimer's patients, as a way to prevent the onset of disease. We have an Alzheimer's prevention clinic. If we see a patient with a bad mutation that's meant to predict that the patient will develop Alzheimer's, we can enroll these patients into protocols to decrease the onset of Alzheimer's. The future of precision medicine is clearly, in our view, also using AI. We're using AI now for all kinds of different things. A lot of medical images at Cornell are analyzed using AI. AI allows us to make very individualized diagnosis by, for example, looking at tumor cells, images of tumor cells, and applying AI to say, well, these tumor cells are bladder cancer, they're breast cancer, they're lung cancer, they're different subtypes. We're using AI across the board, and that's really part of the future of precision medicine. We're using AI also for genome interpretation without going into too many details. Our goal is to make every patient actionable in the sense that there is a mutation that we can use to treat each patient. And just to finish and to connect this, also to visualization, we're very excited to use also data visualization, advanced data visualization as a way to help understand cancer genomes and cancers in general. We can now analyze cancers at the single cell level. We can generate information about millions of single cells for each individual patient, but that's just way too much data that you can analyze, and sort of visualize. By using tools such as augmented reality, and I don't know if I can play this movie, but I guess I couldn't. We can now analyze these tumors in 3 dimensions, looking at each individual cells, what are the genes that are expressed within individual cells, and we can really look at cancers in a very sort of novel way, in a very detailed way, and that's really helping us in terms of finding the best treatment for each individual patient. I'm going to stop here, and thank you.